When skin's healthy, life can be great. But when skin fails, life is always tough. And because skin matters, we want to help patients by discovering the next generation of therapies. We've been making great progress over the last few years, and we're starting to build a strong team. So my background is a drug discovery scientist, working initially in industry at Biofocus, which is part of the Charles River Group now. And then I've been 13 years at the University of Dundee in their flagship drug discovery unit. So our first non-executive director will be Dr. Alan Wise. He's a drug discoverer as well, with a pharmacology background, experience taking drugs into the clinic. But perhaps most notably, he led the company Iomet to a successful exit of around $400 million. The idea is to have a pretty tight internal team, so we'll be mainly virtual with the brains internally, and then tapping into skills and capabilities as needed to support the programs. So our approach to dermatology is that we look for mechanisms that in principle would be effective, but which no one has used in dermatology to date. And then we demonstrate a new use for these mechanisms, so we can obtain a first-in-class position. And then with the biology validated, we then conduct patent busting to get new chemical matter that we can protect in a composition of matter patent. So we don't repurpose, we don't reformulate, we discover new drugs for new indications. At this point, we obviously need to discover the drug candidate we'll take forward. And for oral dosing, this is really around exquisite selectivity and getting the right profile for once-a-day dosing. For topicals, we can benefit from the fact that we can locally apply the drug to skin. And if you apply drug to about 10% of the body surface area, it's very rare to have significant blood levels of the drug from topical absorption. And we can maximize this by modifying the compounds so that they're rapidly cleared when they reach the bloodstream. So we have a very high therapeutic index. So we've applied this approach, which is called soft drugs, to 3 programs to date across different biological targets. We published a little bit around this, but it isn't really our focus. We're product developers. But really, the key marker of our credibility is that both Leo Pharma and Almirall, who are 2 of the world-leading dermatology companies, have chosen to collaborate with us to discover new topical soft drugs. And in the process of working with these companies, we've been able to learn from them and optimize what we're doing and ensure that we have the right processes and practices. So the first indication I want to tell you about is eczema. It's one of the most common diseases to affect people in the world, with about 120 million patients in the developed world alone. And the market's currently going through rapid growth, and it's considered to be in a bit of a gold rush at the moment. So it's growing from about 6 billion market today, which is dominated by topicals, to a market size of around 18 billion within the next 10 years. And it's thought that this has been driven by new product approvals, both topicals and systemics, and such as orals or injectables. So if you have eczema, treatment options aren't ideal. Steroid drugs can be effective at treating inflammation that underpins eczema, but they also can cause permanent skin damage. And so patients are probably too scared to use these drugs in the first place. Tacrolimus, second-line therapy, is also effective, albeit in a different mechanism. But it has issues, which include a transient burning sensation, which does pass but reduces compliance, and it was incorrectly associated with a cancer risk, and although disproven, that reputation has lingered. We've discovered a novel approach to the treatment of eczema that can address both of the biological pathways that are clinically validated. We're very happy to share the biological target on one-to-one basis, but we're not sharing publicly at this time. So we've identified a preclinical candidate topical drug for the treatment of eczema against this novel target. Our drug is very potent against the biological target. It's effective at treating human immune cells and skin cells that are biased to mimic eczema and psoriasis. It's non-toxic in skin cells and there's no liabilities, so no genotoxicity, phototoxicity, and we don't have skin irritation risks in preclinical models. We can penetrate well into human skin in fan cell studies, so this is whole human skin absorption, but we're rapidly cleared from the bloodstream of mice. We formulate it in simple vehicles that were selected in consultation with pharma companies. And we filed a patent in April 2019, so it's defensible. The cell data is all fine and well, but really, if you want to have confidence a drug will work in the clinic, you need human whole tissues. So we've conducted a study where we've looked at abdominoplasty tissue, so ex vivo, and then this is looking at 3 donors, and then 3 replicates per donor, and 3 technical replicates at the back end. So there's 9 data points aggregated into the averages on this chart. So we've compared our drug to ruxolitinib, which is a clinical phase 3 JAK inhibitor. So it probably represents the kind of best hurdle for efficacy. If we can beat a JAK inhibitor, then we're probably onto a winner. And we can see that against the key marker for eczema, which is IL-4, that we have superior efficacy to JAK inhibitors. When we look at other markers, such as IL-13, we're again greatly superior to a JAK inhibitor. So this gives us confidence that we're going to have a high chance of efficacy in the clinic and possibly even superior efficacy to JAK inhibitors, which are extremely effective, but have now received a black box warning for cardiovascular and other safety risks. So we have 6 markers in total, and we can share more data if anybody's interested. We also looked at autoimmune biology. So this is single point data, and again we're looking at the key markers. So IL-17 is the best validated biomarker for psoriasis and autoimmune disease. The biologics that target this biomarker are almost completely effective in psoriasis. So ruxolitinib is not effective at blocking this marker in human skin. Betamethasone is effective, but we are clearly greatly superior to either of those drugs against the key marker for psoriasis and autoimmune disease. Again, we've looked at other markers, and against some of these markers, the JAK inhibitors are quite effective. But across the board, we're generally seeing that we're more effective than a JAK inhibitor in models of eczema or psoriasis. So we've also developed an oral program, which is looking to develop a selective version of our topical. So we have compounds that are highly potent against the biological target, but have around 1,000-fold selectivity against the closest off-target. These compounds are effective again at treating inflammation in eczema models in cells, although we didn't see much of an effect against Th17 biology in the cellular models. We don't have liabilities with the molecule. We think it's developable. There's no genotox or other such issues. And we think it's developable because it's got attractive physical properties, good solubility in FASF models, and we filed a patent last year. But this project does need some lead optimization chemistry. In particular, we need to optimize the oral bioavailability, which isn't good enough to be a single daily dose. So we looked at this lead compound in, again, whole human skin, so the gold standard model. And again, we're seeing really good efficacy against the key marker of eczema, which is IL-4. And we've got comparable efficacy to a potent steroid and around a sub 100 nanomolar EC50. So we're pretty pleased with this. What we were slightly more surprised by is that we're also seeing an autoimmune effect, which wasn't predicted in cellular models. So this is probably something that people aren't going to stumble across unless they do the whole tissue models that we have done. So we looked at 6 markers, and across all of the markers of autoimmune disease, we have comparable efficacy to a potent corticosteroid. So if you think about this from a clinical perspective, if you're in crisis with asthma or MS or pretty much anything, the first thing doctors are going to give you is oral steroids. So if you can show that you have comparable efficacy to an oral steroid, you've probably got something pretty special. So I should mention that we've just learned that the selective compounds have the ability to have a 24 therapeutic index. So we have the safety aspects as well as the efficacy now to justify doing a lead optimization project. So when skin fails, life can be pretty horrific. If you've got a genetic skin disease, you're living in constant pain and discomfort, and probably going to have a shortened life expectancy. And we're particularly interested in recessive dystrophic epidermolysis bullosa. So patients with this disease lack a key protein that forms a skin barrier, and as such, the skin basically just falls off at even slightest contact. It's very rare, but because of the extreme medical need, the cost of care in the US is estimated at around $100,000, and this is without any disease-modifying therapies. So this is Jackson, he has EB, And as mentioned, his problem is he lacks a protein that forms an anchoring tendrils in skin that hold the skin together. And this protein is called collagen 7. And without it, there's literally nothing to hold on to. So around 30 to 50% of cases of recessive dystrophic EB are caused by premature termination codons. And this is where the body produces a truncated, non-functional version of the protein, so it's almost complete knockout of the gene. There's clinical proof of concept that you can restore the full functional collagen 7 with a drug called gentamicin. So gentamicin is able to cause the body to ignore these mutations. And what the authors of this study did was they injected the gentamicin into the dermis of skin, or they applied it to open wounds. And they saw an almost total restoration of collagen 7. Really excitingly, that collagen 7 remained for 3 months after treatment stopped. So it's a very long-lived protein in the skin. Unfortunately, gentamicin is not the ideal drug. It's toxic, you can't give it orally. It's very synthetically complicated, so you can't make better versions. It doesn't penetrate skin at all because it's very polar, and it's also a very weak read-through compound, only functioning at about 3 millimolar level, which isn't, it's pretty high. So, so I'll skip over this. So we conducted a screen to identify new compounds that could do read-through. And we've identified chemical matter that's over 1,000 times more potent than gentamicin. Our compounds have a structure-activity relationship, so it's not just one hit, this is a series of molecules with consistent biology across multiple cell contexts. We've got sub-micromolar leads, and we've now shown that one of our analogues is highly skin penetrant and is able to achieve a 3 millimolar level in skin. We estimate this is 300 times the effective concentration. We also think that this project is as close to unique as you're going to find. So we know this because we collaborated with big pharmaceutical companies and screened their compound libraries, and we still have the best compounds in hand. So we didn't discover anything that was better. And many other companies and biotechs have tried to crack read-through. It's such an obvious target to go for, but no one has been able to do it. So excitingly, we can also show that our compounds are able to produce collagen 7 in patient-derived cells. So when we treat the cells with one of our compounds, we can see about 20% restoration of collagen 7. Notably, gentamicin that I showed in the clinical proof of concept is not effective in this study because it's such a weak read-through compound, even at very high concentrations. Geneticin is an analogue of gentamicin that's much more potent, but is very toxic. But in the cells, we do see restoration. So this shows that the cell model's behaving normally. This isn't aberrant data. And that our compound is comparable to geneticin at high dose. This is very exciting because you only need about 1 to 2% of collagen 7 to be therapeutic. And we know this because there are missense mutations where patients have about 2% of those anchoring tendrils in the skin, and their disease is much milder than patients with premature termination codons. So we think we've got a great competitive advantage because there are no systemic drugs in development. And of the drugs that are in development, they are either only symptom modifying or they must be applied to open wounds or injected to every site in the body. So we're the only drug which you could apply to any skin site and which would be disease modifying. What's really exciting about this program is that beyond EB there are many indications this same molecule could be therapeutic in. So once we've delivered proof of concept in epidermal bullosa, we can expand into other high need indications. So dermatology is attractive in terms of pharma deals. So I've just kind of cherry picked as you do. But if you look at discovery stage oral therapeutics, raw gamma deals were achieving valuations of up to $500 million in a discovery stage deals. And on average, 3 programs that were licensed at this stage were around $230 million. So oral anti-inflammatories of high interest to companies. In phase 2, Zircon was acquired by Novartis for a speculated $1 billion after phase 2 proof of concept. Leo Pharma acquired a topical orphan disease program for $750 million after a very small phase 2 clinical trial. And Crisaboro was acquired by Pfizer just as it was about to get regulatory approval for $5 billion. So dermatology is a highly commercially attractive area to be developing drugs. So just to summarise, we're an experienced team of drug discoverers and entrepreneurs, and we're really focused on commercial exit. So we're really driven to have a commercial output at the back end of this. We've got a portfolio of really attractive projects that spread the risk across multiple indications of high need, and we're targeting a rate of return of at least 20x for investors. So thanks for your time and attention. I'm very happy to answer any questions you might have.